this is the Ahmed Ali, the second presentation. You can introduce. İbrahim Hocam. Sesinizi açın. İbrahim Hocam. Sevgi Hocam siz sunum...
Duyuyor
musunuz?
Okay.
Tamam.
Duyuyorum ben.
Okay. Thank you, Dr. Şekeroğlu. First of all, I would like to thanks to Dr. Şekeroğlu and his team. Special thanks to for kindly and nice symposium. This symposium is very important. This MESMAP. Today is we will start sixth symposium. I think is it true? I think.
Yes.
Yeah. Okay. Again, good morning. Now, this presentation belongs to Mr. Malik and Mr. Ahmed Ali about preventive of advanced antiglycation products by the seed extracts of Nigella sativa. Please, Mr. Ali.
Ahmed Ali.
Please,
Yes,
Mr. Ahmed Ali.
A very good morning to all of you. And thank you, Professor Dasim and your team, Dr. Sevji, the secretary of this conference. It's an honor for me to be a part of this MESPAP. It's third year. I've been attending. I started my first presentation and attending this conference in 2018. That was MESMAP 4. We were looking forward, but nature has its own. So anyways, I wish all the best to Professor Nazim and all the members of organizing committee. And last but not the least, I'm thankful to Professor Nazim for giving me this opportunity to present my work. There is slight a spelling mistake in the title of this. The title of this my presentation
you
is...
have presentations?
Yeah, I will share.
Please share.
So, is it visible now, Professor? Okay, is my presentation visible? Yes, thank you, Professor. Okay, so the title of my presentation is Prevention of Advanced Glycation End Products by the Seed Extracts of Nigella sativa. The common name of this seed in English is black cumin, and it is a very well-known herbal medicine in Arabs and all over the world. They have been used for different purposes, but my focus is on the biochemical events which happen during the diabetic process. So just a brief about, because we are all concerned about this diabetes event. It has become a major metabolic disorder or non-communicable disease, and almost by another 20 years or 30 years, 50% of the population will be residing in developing and developed countries. So basically it involves the impairment of glucose metabolism. I'll not go into detail of this, what happens during the pathophysiology of this disease, but the modern theory of diabetes suggests that it is an implication of oxidative stress and glycation. And because of these modifications in the cellular systems, there are many secondary complications which are associated with diabetes. So glycation is a non-enzymatic process which happens between the sugar and any other molecule which has a amino group. Okay. So this is a non-enzymatic reaction. To differentiate it from a enzymatic equivalent, that is glycosylation in the endoplasmic reticulum, and Golgi apparatus. So there are several stages before this reaction is complete: early, intermediate, and late stage. At the end of the reaction, a group of molecules are produced, which are known as advanced glycation end products, which have been implicated in many diseases because of their capability of changing the structure of biomolecules. So you can see the scheme here. Sugar and proteins react, and in the initial stage, a CIF base and amadori bases are produced, which further undergo modification, and dicarbonyl compounds are produced. And finally, you see advanced glycation products, which alter the macromolecules. So we can see that hyperglycemia leads to an increase in the ages, and these increases alter the molecular structure. There is another pathway by which ages work. They bind to their receptors on the various cells, and they increase the expression of nuclear kappa B, nuclear transcription factors like NF kappa B, ultimately leading to an increased oxidative stress, and finally causing a group of disorders like Alzheimer's and nephropathic and retinopathic and cardiovascular diseases. So people have been looking for this inhibitors for this age, and some synthetic compounds have been synthesized, and some natural compounds are being looked for. So these inhibitors can interact or can inhibit the interaction between the sugars and proteins, or they can inhibit the formation of amadori products, and they can inhibit at later stages. So among the natural compounds, plants and among the natural inhibitors, plant extracts and formulations are being studied. This is the cellular mechanism by which toxicity of glycation is suppressed in the systems. We have enzymatic systems like aldehyde reductase, glyoxylase, amadoriases. We have protein degrading systems, vitamins, uh, synthetic compounds like aminoguanidines, and world has been looking for anti-glycating agents and antioxidants from. So my focus is to characterize the role of Nigella sativa seeds in the process of glycation. We all know that these seeds are quite rich in carbohydrates, proteins, amino acids, and volatile and fixed oils. One of the major phytoconstituent of this uh, plant is, or the seeds of this plant is thymoquinone, which possesses various activities like antimicrobial, anti-tumor, antiviral, anti-inflammatory, anti-diabetic, and so on. So the structure, these are the four, some of the major phytoconstituents. As I mentioned, that one of the major constituent of this is thymoquinone. We have recently produced, I have recently published a review article on this, where I have mentioned in detail about the properties of thymoquinone and Nigella sativa. So what we have done, we have collected the seeds, and then we have prepared the plant extracts, maybe aqueous or methanolic, depending on our requirement. Then we did antioxidant assays like DPPH, where we did alpha amylase inhibitory assays for anti-diabetic properties. That was that was initial characterization. We also did phytochemical characterization.
Excuse me, Mr.
Yes.
Ali. I would like to say sorry. Our time is very important. This on the symposium.
Okay.
Time,
Yeah,
time is shortened. Last 5 minutes. Okay.
Yeah, I'll take it.
Please.
Yes. Thank you, Dr.
Please.
Ibrahim. I'll take it. Yes.
Thank you for your kindly.
Yeah. So these are brief phytochemical analysis. We can see that total phenolic contents and total flavonoid contents, and the alpha amylase inhibition was almost 50%. Similarly, we could show that increasing concentration of aqueous extract inhibited the free radical generation. I'm coming to now glycation, where I can show that the browning and fructosamine content, the early glycation products were quite significantly reduced in the plant extract. green line shows the control, the glycation system, and the red bars shows the treatment or extract along with the control system. So we could significantly show that the early glycation products and Maillard reactions are significantly inhibited. Then we measure the advanced glycation products and glycation-induced aggregation. Here also we showed that the inhibition was quite significant in the presence of Nigella sativa extract. We measure the total amount of advanced glycation product using spectrophlorimetric observations. We could show here also that Nigella sativa extract was quite efficient in reducing the total amount of ages. We did glyoxidation. We checked the effect of Nigella sativa extract on DNA damage, and we could show that Nigella sativa extracts were able to prevent the glycation mediated DNA damage. This work has been published in the first issue of CupMap. We have also worked on thymoquinone, the phytoconstituent of Nigella sativa extract, where we could show that thymoquinone was also very efficient in inhibiting the browning, fructosamine content, and carbonyl content. We could also show that uh, aggregation was also significantly reduced by this Congo red and thioflavin T assays. The total ages was almost reduced by 78% by a higher concentration, that is 20 micromolar of thymoquinone. Then we did an fluorescent quenching study where we showed that there is an interaction between the BSA and thymoquinone that is very important when you are going to develop a drug. So we could show that increasing concentration of thymoquinone was more able to quench the absorbance of BSA. We did the similar kind of studies with the thymoquinone, which we had done with Nigella sativa, and we could show that thymoquinone was also very efficient in reversing the strand breakage of the DNA. So some of this work has been published in one of the journals of natural volatiles and essential oils, where we have shown the anti-glycating and anti-aggregation potential of. So with all these experiments and results, we could establish that the Nigella sativa was quite efficient in inhibiting the production of early and advanced glycation products. It was also able to prevent the glycation-induced aggregation, which we checked with the aggregation of proteins. The black cumin seed extract was also able to prevent DNA damage, which indicates its role in as both or the role in preventing the oxidative DNA damage may be a combination of anti-glycating and antioxidant potential of black cumin extracts. And our preliminary studies also suggested that alpha amylase was inhibited by extracts. One of the major significant findings was that thymoquinone was more potent as compared to the extract in preventing the formation of glycation products. So the conclusion of my study is that the aqueous extracts of Nigella sativa seeds clearly show anti-glycating, anti-aggregation, and this property may be because of antioxidant property also, because now people are suggesting that this antioxidants also can work as anti-glycating agents, because one of the mechanisms by which glycation works is to generate the free radicals. So these seeds maybe can be further characterized and can be used for significantly preventing the secondary complications of diabetes like glucotoxicity, can be used for preventing the protein aggregation mediated neurological disorders, and pure compounds can be used to develop drugs on the basis of them. So this work has been mainly done by two of my students, Prerna Malik and Dinesh Kumar. I have received fundings from various funding agencies, and my collaborators are this. Thank you for giving me this opportunity. And if any question, please.
Thank you, Mr. Ali. Should have any question? No. Okay. Please, Mr. Şekeroğlu.
You hear me? Have you hear me?
Yes. Yes, I
Mr.
can.
Ali,
Yes.
thank you
Yes.
for your nice presentation. You know, our Prophet Hazreti Muhammad said that Nativa Sativa, Nigella Sativa have cure all disease. You should
Yes.
use the seeds mostly care, you know?
Yes.
Yes.
It cures for everything but death.
Okay,
Yeah,
okay.
yeah.
Thank you for your
Thank you.